Okay. I will start now. Okay. I'll start now. I would like to thank the organizers for giving us the opportunity to discuss our work today. We'll have 3 short talks, one by me and one by Arijit and Zeljka. My name is Professor Marija Krstic-Demonacos, and I did my undergraduate degree in and master's degree in University of Belgrade in Serbia. I did my PhD in University of California, San Francisco in the States, where I researched steroid receptors. And my postdoc was in University of Glasgow, where I investigated p53 tumor suppressor. I joined University of Manchester as a Wellcome Trust Fellow and lecturer in 2003, and University of Salford as a professor in molecular medicine in 2013, and as a director of Biomedical Research Center in 2014. I'll first provide an overview of biomedical research in the University of Salford, then summarize quickly my research interest and focus on one project at the end of the talk. I'll share my screen now. Can you see this? No?
No, not currently.
Right. How about now?
That's great. Thanks, Marija.
Okay. Right. So key people in the school are our new head of school who joined recently, Joe Sweeney, and 2 ADs for research, Dr. Hadi Mostarzadeh and Dr. Ian Goodhead, and myself, who lead Biomedical Research Center. You can have here the link to our website if you're interested to know a bit more. There are 30 researchers in BRC in University of Salford. We work on range of biomedical and health topics, from cancer stem cells to pathogenic microbes. BRC is part of School of Science, Engineering and Environment in University of Salford, and has 3 main research areas: cancer diagnosis, prevention, cardiorespiratory physiology and pathology, and infectious diseases. We also have research in dementia and have chemists who help us with all these 3 areas of research. We have invested multi-million pounds into the infrastructure to create polymer and translation medicine laboratories, where we can do various different things listed here, including physiological recording of the heart cells, CPS disc centrifuge for nanoscale characterization, cancer metabolism suit, and a range of techniques for the chemistry, immunohistochemistry, microbiology, parasitology. The cancer group in BRC is the larger research group. There are various different research areas listed here. If you're interested, you can have a look at the BRC website. We are interested in inflammation, steroids, p53 tumor suppressor, cancer stem cells, aging, biomarkers, nanobiotechnology. Quite a few of us focus on childhood cancers. And we have a group of chemists who design and synthesize novel compounds to treat cancer, to test them in various different cancer models. Now, this is not, okay. My research interests are cellular response to stress, how cells respond to stress. And there are actually, I focus my research mostly in 2 different areas. One is response to steroid hormones, mostly glucocorticoids, and the other one is cellular response to DNA damage, focusing on p53 tumor suppressor. When we talk to about the glucocorticoid hormones, they're hormones of stress. Main hormone in our blood is cortisol. Glucocorticoid hormone is cortisol. So when we are stressed, in our brain, hypothalamus secretes a corticotropin releasing factor that then affects pituitary to synthesize adenocorticotropic hormone that then causes adrenal to synthesize cortisol, and then cortisol allows responsive cell to stress. And obviously we don't want to be permanently stressed out, and there are several feedback loops to ensure homeostasis. The steroid hormones are small lipophilic molecules. They are synthesized from cholesterol. Glucocorticoid hormones play an essential role in response to stress. They regulate metabolism, development, and they are very important because they suppress immunological and inflammatory responses. They are tightly regulated in the blood. Excess can lead to syndromes such as Cushing syndrome and deficiency to Addison disease. They are amongst most commonly prescribed drugs, and they're used in treatment of a wide range of diseases. However, because of the side effects, we want to know more about them. This is oversimplified mechanism of action of glucocorticoid hormones. Glucocorticoid receptor is activated by glucocorticoid hormones that enter the cell and in the cytoplasm bind to glucocorticoid receptor that then dissociates from chaperones, and then glucocorticoid receptor hormone complex goes to the nucleus where it binds response elements to activate or repress transcription. The levels of regulation of glucocorticoid receptor are indicated here. They are depend on the cell type, ligand, GR is post-translationally modified, and its stability is also regulated, and there is very large number of cofactors that also regulate glucocorticoid receptor. My research started in Belgrade, where for my master's I was interested in studying steroid receptors in cancer. That continued in San Francisco, where I mapped the sites of phosphorylation on glucocorticoid receptor and kinases involved. In Manchester, I continue to work on glucocorticoid receptor and its interaction with TTC5 cofactor. And I found the link in between phosphorylation and SUMOylation and control of receptor activity. In University of Salford, my interest continued and was focused on childhood leukemia, how glucocorticoids are used to treat childhood leukemia. Because they are the drugs of choice, dexamethasone, prednisolone. They kill white blood cells, and mechanisms of resistance, especially microenvironment role and autophagy role, and mechanism of resistance to glucocorticoids, as well as bioinformatic approaches, were focused on my research in Salford. The other aspect of my work is a p53 tumor suppressor and its role in cancer. Mostly because it is such a pivotal molecule in cancer. It's a transcription factor that's mutated in more than 50% of human cancers. It's called the guardian of the genome because of its crucial role in protecting genome. It is activated in response to stress, function as a cellular brake. It stops cellular proliferation, so the cell has time to repair the DNA when it's damaged, or if it is damaged beyond repair, it facilitates apoptosis or cell death. 53 pathway mutations and alterations are found in cancer, and because of that, the uncontrolled division results in tumor formation. There are various different levels of p53 activity control. It is in normal cells quite unstable protein because it's degraded by MDM2 ubiquitin ligase, and it is stabilized by DNA damage, and then it controls a wide range of processes indicated on this slide. It is heavily post-translationally modified. Post-translational modifications control its activity. I continue working on p53 in Glasgow, where I isolated TTC5, a cofactor of p53. And in Manchester, I was interested in bioinformatic approaches to understand p53. And this is the project I will spend the last 5 minutes talking about. I'm not a bioinformatician, but this is my favorite project, and I will try to explain it as best as I can. This is a long-standing collaboration between me and Jean-Marc Schwartz, Krstic-Demonacos in Manchester, Emir Bakr in Yukon, and Kun Tian in China. So most cancers are treated with surgery or radiotherapy and chemotherapy, and radiotherapy and chemotherapy affect p53 pathway. However, responses differ, and more we know about these associated p53 pathways, higher the chance of us understanding what happens in the tumor. Therefore, we decided to apply a bioinformatic approach. Scheme is indicated here. So what we did is we extracted information for string protein-protein interaction database, and then created logical model that was then analyzed, validated, and tested to provide predictions. So the aim was to investigate DNA damage inducible pathways to cancer, role of tumor suppressor in its building logical models. And the reason for it is because there are over 100,000 papers about p53 in PubMed, therefore making it very difficult to study. There is on the other side, large number of clinical studies that report changes in the genome or transcriptome and proteome from cancer patients. And in this project, we try to integrate these two types of data. And we hope that we can analyze pathways dynamics and superimpose it to genomic profile and clinical symptoms. What we created was a logical model, Boolean network, indicated here. We used Java-based program to retrieve the information from string, made the network analyze it using CellNet analyzer, and visualize it using Cytoscape. So here what you see is nodes that represent genes or proteins, then input, DNA damage, or output apoptosis, or senescence. And each line here indicates activation or inhibition. So what this model can do, using various different simulations and algorithms, you can turn the input on this model, mimicking DNA damage or chemotherapy, remove the node, mimicking mutations in cancer, and analyze how the whole network changes because of these either mutations on or having DNA on or off, using something called dependency matrix that defines differences before and after the mutation, or algorithms that can give you a certain information when you compare predictions of the model simulation to omics type of patient's data. What do we hope to achieve, and what we have achieved so far? Well, we have had our proof of principle that we can create a large model that has a predictive capability, and they can generate novel knowledge how DNA damage affects cells and how we can follow certain pathways in the actual patient's omics data. We also learned that feedback loops and ambivalent effects play an important role in actually providing stability of the cell to external perturbations. What we want to do, we are seeking collaborations. We are actually actively talking to Dave Wedge in Manchester University to continue with this model and try to obtain the funding for. This model can be adapted for particular drug or protein or cancer type. And what we want to do is design interface to actually automate a comparison of data from patients to model predictions. We want to build a database for it. This model is classed as medical device and patented. And we want to test the model across a wide range of cancer types because we want to train it for the highest predictive power. We propose to use machine learning approach for this. And we want to identify the shift in pathways after therapy. So the problem with cancer is that once you apply the chemotherapy, the resistance occurs, cancer comes back. And if you can continuously test and redesign the therapy using the predictions of the model, then you can potentially have a vision of turning cancer in chronic and not terminal disease. This is my lab, past and present, the funders, and we sometimes have fun as well, in addition to working hard. Thank you very much. How do I get back now?
Okay, I will just share my screen. That will help. I think.
Yeah, thank you.
No problem.
Okay.
Can you see my screen?
Okay, can I introduce Zeljka then?
Thank you.
All right. Zeljka is, do I have your, or would you like to introduce yourself? It's better.
Yeah. Can you see my screen? Fine.
Yeah.
Yeah. Okay. Thank you very much, Marija, for your handover. So my name is Zeljka Krpetic. I'm a lecturer in physical chemistry at the University of Salford. And I joined Salford in 2016 after a very long journey of being a postdoc across UK and Europe. So I worked in some amazing places, such as University of Liverpool, University of Strathclyde, Centre for Bio-Nano Interactions at the UCD Dublin. And I acknowledge a variety of knowledge that enriched my initial journey when I started as a master's student and a PhD student at the University of Milan in Italy, so a long time ago. In my research, I focus on nanofabrication. So what we're mainly doing is nanoparticle synthesis, functionalization with different drugs for applications in cancer nanotechnology and also antimicrobial applications. So today I'm going to bring you on a journey of how do we characterize a nanoparticle before we apply them in any of the settings of the nanomedicine, which is a very important point, which not everybody is considering when using the nanoparticles when applying them in vitro or in vivo. I will talk about some challenges, and I will talk about some cutting edge things that we did, which is quite important in understanding the bio-nano interactions. So where the nanotechnology is now is that we're at a really good point in implementing these objects at a nanoscale, very small scale between 0 and 100 nanometers in living systems, but we still have challenges in crossing biological barriers in particular and overcoming biomolecular corona formation. So I'll talk about that a little bit today as well. So in my group, we make mainly anisotropic nanoparticles because we are studying how the shape affects the biology, the different aspects of it. And mainly we're trying to find systems that overcross biological barriers and avoid endocytosis. So there are different approaches that we are using. I'm not going to talk about this today. When we are at the point of nanofabrication, what is really, really important is a batch-to-batch reproducibility and making sure that nanoparticles are monodispersed both in shape and size. So what can happen to nanoparticles, no matter how we make them, what method we use, they can undergo aggregation if they are distributed in biological media, and that can lead to either withdrawal of a particle from the system, so you can have reduced dosing of your nanoformulation, or it can lead to unwanted cytotoxicity effects if your particle size distribution is very different. So there's a lot of things that need to be done around particle characterization in order to bring them to the stages of clinical trials, etc. So not only from the physical chemical point of view, but we need to focus on the characterization of biomolecular coronas and the particles in biological fluids, and also how do we translate the biological methods into in vitro and in vivo testing. In my research, I mainly use gold. So gold is an excellent material because it offers lots of flexibility around monodisperse synthesis, both in shape and size. It's excellent. Contrast at the microscopy allows us to view where the particles go within the biological cells as well, and it can be used also in sensing and diagnostics. In order to create very stable systems that are relevant to targeting different diseases, we are using specific surface chemistry approaches in order to link the drugs or other moieties on the surface, so we create the particles that are with a very well-defined surface chemistry. So this is just a small table that I came up with what we need in order to characterize gold nanoparticles to give us a full picture of what happens to them during the fabrication process after functionalization, and also what happens to them in biological systems. So I'm not going to go into details of these methods. These are just a few that I could come up with as a postdoc, but what I'm focusing mostly today in this talk and in my career is differential centrifugal sedimentation. So this technique in particular, I was introduced to it a long time ago, around 2007, when I was a postdoc in Liverpool. We were developing methods to characterize particles that are functionalized and understand how we can study these particles in situ, what happens to different distributions. So it is a bench-sized centrifuge. It's an analytical centrifuge, not preparative, that separates the particles by their size when they're sedimenting under very high speed under biological, under the density gradient. What this does, it allows a high definition resolution sizing, and what we were able to do using, for example, some target ligands, we were able to distinguish ligands that differed but just one methyl group. We were also able to predict if the peptides that we were using on the surface had a bound structure or they were linear. So really high definition sizing. So how did I go ahead and apply these? So we were able to also understand small changes in nanoparticle aggregation. And if you have, for example, formation of dimers and trimers as part of the sensing strategy. So this confirms the TEM picture. Talking about biomolecular coronas, if nanoparticles are introduced to biological media, they undergo the coating that's called biomolecular corona. It's a dynamic layer of biomolecules, not only proteins, but mostly proteins that are affecting the functionality of a nanoparticle. So this sometimes can pose challenges in particle uptake and the way the particles interact with cells. With using gold, it's quite a challenging thing to understand the particle size because it's counterintuitive. So when we are coating a gold particle with a protein, the diameter appears smaller. So this is not because the particle becomes smaller, but it's because the whole object becomes lighter. So the main message from here is the method cannot be used just to extract the final diameter, but we can monitor what happens to distribution after certain interactions. So we can use this to predict stability of particles and measure the soft coronas. After incubation with cell culture medium, for example, we can see some formation that is linked to these shifts. We could also use this to study the particle surface chemistry, so how the particle surface chemistry affects biomolecular corona formation. In a more complex scenario, I'm not going to describe this study in the remaining 2 minutes, but we were able to actually design a study to understand and quantify the number of proteins that are attached to model particles that we use to prove the kinetics of binding and epitope mapping of these particles. So what we did, we took an immunogold approach. So immunogold is a very small gold nanoparticle that contains an antibody attached to it. So the hypothesis is that each gold particles that we see here represents one particular antibody. So if we take a model particle, in this case it is polystyrene, about 200 nanometers in size, and we coat it with a pre-designed corona, we will be able to map out the exposed epitopes. So we also use some complex microscopy imaging and theoretical modeling in order to piece out this system. And it's a really good system that can make sure that we can quantify not only the number of proteins, but we could also understand how these are distributed in a live corona. So if we have also in the case of plasma fluids. So everything I described here was in accordance with the theoretical predictions, and this method in particular was used to profile the biomolecular coronas. So I hope you've been inspired by this journey. We also recently published a study that describes some real challenges in characterizing extremely small nanoparticles, in particular those coated with polymers, because the shift of the DCS can go either way. It is just it's a testimony that a lot of new studies can be done, and this is the path that we are pursuing in my lab. We're currently analyzing nanoparticles that are anisotropic, containing biomolecular coronas, and also hollow particles that are a new challenge for me. I would like to thank my research group, so Nanolab at Salford. We are funded by KidScan and DTA for the moment, and running for some other funding with colleagues in Manchester University, Professor Fred Curran in particular, and I'm also working with the scientists from the University of Liverpool, and hopefully you'll be able to hear about our new studies next time. Thank you very much for your attention.
Okay, thank you, Zeljka. Arijit, would you like to introduce yourself and share the screen?
Yeah, thanks, Marija. Hope my slides are visible.
Yeah, we can see them.
Yeah. Thank you. Yeah, so thank you, Marija. Thank you to the organizers for giving me a few minutes to talk about what my group does at Salford. So a little bit brief about me. I've also joined, similar to Zeljka, in Salford in 2016. Before that, I was a principal investigator and a group leader in a genomics institute in India for about 9 years. So that's my career path. And before that, I was a Marie Curie fellow as a postdoc in Netherlands. And before that, I did my PhD from India in human genetics. So today I will talk about how we use RNA as cancer biomarkers. Because of the time limitation, I will not be able to talk on all the aspects that we do in this space. So this is the outline, what you are going to hear. So I will talk specifically about microRNA, which is one of the branches of what we do. And I'll talk specifically of a specific set of microRNA present in the human genome called microRNA cluster. And I'll briefly tell you about what is so special about this specific cluster. So why do we choose to work on this cluster? And we'll show you what we found about biomarkers from this cluster in tissues, mostly from brain cancer, but also a little bit from other cancers I'll show you. And I'll talk very briefly about what my lab is currently trying to do and the future possibilities and collaborations. So very brief background on microRNAs. MicroRNAs are one of the major type of non-coding regulatory RNA. So non-coding means these RNAs are functional, but they do not make proteins. What you are seeing on the screen is that this boundary here is left of that boundary is nucleus of a cell, right of that boundary is a cytoplasm of a cell. And like any other transcription, microRNA genes also get transcribed by RNA polymerase II. It's regular transcription, but then it has a stem loop structure like you see here. And then within the nucleus, it undergoes one round of enzymatic cleavage. And then this structure here comes out in the cytoplasm and it undergoes another round of enzymatic cleavage. And then this loop goes away, then it remains this kind of structure. Then one of the strands of this one of the strands, which is about 20 nucleotide long, gets incorporated as part of a protein complex. And then they usually bind to the 3' untranslated region of mRNA. And the result of that binding is usually downregulation of the transcript. So the expression, if a microRNA binds to a mRNA, then the expression of that mRNA usually goes down, right? So it would be important to remember that. And in this binding of microRNA mRNA pairing, the most important is 7 nucleotides here that is called a seed region. So this will become relevant to the rest of the talk. So please try to keep that in mind. So we will be talking about a particular cluster in chromosome 14. In brief, we call it C14MC, so C14 microRNA cluster. This is the cluster right here, which you can see outlined. And this is part of a domain of about 1 megabase pair long on the genome, on the DNA, on chromosome 14, where it has multiple protein coding genes that is indicated by either blue or red, or non-coding, different non-coding RNAs. Here is one block, here is one block, here is one block. When I say imprinted, what I mean by that is depending on the parental origin, it may or may not express. So for this particular microRNA that I will be talking about most of this talk, they are expressed on the copy that you inherit from your mother. They are not expressed on the copy that you inherit from your father. So half of your copies express, half of your copies don't express because of an epigenetic regulation of this entire domain. So but classically that's the definition of imprinting, that there is a parental origin difference in expression of this RNA. Why we want to work on microRNA clusters? What is the interest about clusters in general? The interest is, if you think of, this is the DNA scenario here, where a cluster means there are multiple microRNAs sitting next to each other, and it will have a promoter that will transcribe this as one long piece of RNA from where individual microRNAs will be processed. And these individual microRNAs will come into the cytoplasm. Let's say what you see in the schematic here, I've drawn 6 different microRNAs, this 6 from here, 1, 2, 3, 4, 5, 6, and each of them will bind to, let's say, 6 different mRNAs. And as I told you, the binding result will be mRNA will go down. So the reason we are interested in microRNA is, if let's say all these microRNA go down, if this promoter regulation changes, expression changes, all of them go down, then as a result, all the mRNAs will go up. So by regulating this one switch, which is a promoter, you can essentially part of a whole pathway. And we wanted to test that hypothesis. Before that, we analyzed thousands of patients' data, actual patient tumor data from Cancer Genome Atlas. And this is a snapshot of 12 different human cancers on the left. And on the top, the height of the bar indicates how many microRNAs are perturbed, either up or down. This is the cluster we are talking about. Green means downregulated, and the size of the circle means how significant is the downregulation. The bigger it is, the more significant it is. What you see here, the several cancers, more than half out of 12, this whole cluster, all the microRNAs, there are about 70 odd microRNAs in this cluster. Most of them are downregulated significantly. There are a couple of cancers, lung cancer specifically, where there is upregulation. So what we found is, out of 12 cancers, in most cancers, this cluster does show a coordinated perturbation, mostly downregulation, which means the corresponding mRNAs will be upregulated. So having found that, we wanted to put that on a pathway. So this is the standard RAS P53 pathway that is common in most cancer. This is a pathway specific for glioma, brain cancers. The squares are mRNA protein coding gene, and the circles are microRNA from this cluster. Whatever is in green, we found it to be experimentally significantly downregulated. And so this is how it maps. So like I told you by the schematic in the beginning, that if there's a coordinated regulation, then multiple microRNA from a cluster can regulate one pathway. So this is the proof of that hypothesis. And then when we looked closely, I'm showing you example of 2 microRNAs here, namely 134 and 485-5P, just to specify 2. We have more like that, where 134 has 8 different mRNA, targets 8 different mRNA on this cluster, and 485-5P targets 6 different mRNAs on this cluster, indicated by red stars and blue stars. And as a result, if you look at the survival plots, so very briefly again, the blue line shows that a higher expression of this microRNA results in better survival of the patients. So a downregulation, which is indicated by green, would result in a poor prognosis of the disease. So in other words, they work as a biomarker, where if you find a downregulation, you can relate it to prognosis and different stages of cancer. We validated this hypothesis both in-house and in large-scale bioinformatics data, where we show, so you can't really read on this slide, just on a overall view, just understand that the blue or the green color is for cancer patients and the red is controls. And most of the mRNA, these are mRNA, which should be upregulated. You can see that both in the Cancer Genome Atlas data and our own data, the cancer patients have upregulation of the corresponding mRNAs compared to controls. So there's a validation that most genes in the pathway are indeed upregulated. As I told you that we can link this to prognosis. So these are 2 examples, one in breast cancer and one is brain cancer, that increasingly you see more and more downregulation when you correlate it with the severe form of the disease in both forms. And we have also have this in more cancers now. I'm just showing you 2 examples where these clusters can work as a biomarker, not only for diagnosis, but also for prognosis. Hence we are interested to go further in this cluster. We have also shown in case of brain cancer that the editing in microRNA, editing is a tissue-specific feature in microRNA. And in brain cancer, indicated by GBM, we have shown that compared to normal brain from the brain tumor, the amount of editing goes down, and it correlates with the enzyme ADA2 that is responsible for editing. So not only microRNA expression, but the sequence variation in microRNA, which is a post-transcriptional modification, can also be used as a disease-specific biomarker. This is shown in brain cancer, but again, we have seen this in other cancers as well. And this is where I want to bring you back to this image that I showed you earlier, that the seed region, so most of this editing that I'm talking about happened in these 7 nucleotides, which is crucial for this pairing. So if there is a change in this nucleotides, then the pairing gets disrupted. Hence, you see a dysregulation of the corresponding mRNA. Currently in my lab in Salford, we are trying to identify these microRNAs in the extracellular space, in exosomes. So what are exosomes? Exosomes are biological nanoparticles, these small, tiny balls that are moving from one cell to the other. We are now looking into these tiny balls. If I zoom in, why are we looking into them? Because they carry biomolecules in them, particularly microRNA, because I'm talking about microRNA. And what is the interest? Because these are so tiny, they can cross different biological barriers, such as blood-brain barrier. And so you can find a non-invasive route to look at these biomarkers in cancer, which is easier than for the patients. It's not invasive for them. So these 3 bright PhD students currently in the lab, they have now set it up. So what I'm showing you is a microscopy image of these vesicles, these tiny particles, and we can quantify their distribution. We can characterize them by Western blots. And very early, these are all unpublished data. We show that indeed the RNA comes from the vesicles and not from anywhere else. And this is a very brief data for lung cancer, where we find vaping specific biomarkers from this particular cluster. And we can find biomarkers for vaping related lung injury, distinctly different from other lung cancer types. So this is the initial proof of concept that we can use this cluster microRNAs as biomarkers, not only in tissues from cancer patients, but also in the extracellular space, leading towards non-invasive diagnostics. So that's pretty much the end of my talk. The data I showed you, most of the data I showed you comes from these 5 publications. I'm only showing you the publications that relates to this particular presentations. There are other lines of work in my lab that I'm not talking about today. So these are the 5 publications which I mainly based today's presentation on. Obviously, this kind of project needs a lot of support from colleagues and funding. So I have been funded generously from various public funding organizations from India. These are my collaborators in India. These are my collaborators in Salford. One of the key ones, there are more. And I've been funded also in the UK. And I have been also funded by UK-India initiatives together. So thank you very much for listening. I'll be very happy to answer any questions you might have.
Thank you. Arijit, any questions? Okay, any questions? Okay, if there are no questions, thank you very much for participating, and we can close the meeting. Thank you.